Good morning students. Today we will learn about the phases of cell cycle. Now the cell cycle means the division which takes place when cell divides into two daughter cells. Why it is called as cycle? Cycle means the when anything takes place in a regular manner the cell will divide. When the cell divides, it always prepare itself for the division, and that division consists of two phases. First phase is called as interphase and another phase is called as M phase. M phase is also called as mitotic phase, in which the multiplication of the cells takes place. Basically this is also called as where the actual division of cell takes. It simply means when we go for the exam, it means this is our examination day, in which in 3 hours we give our best, and whole year we prepare ourselves, and that preparatory phase is called as interphase. Now basically just I have told you that in M phase actual division will takes place. It means this is the event which takes place very quickly. Let's take example, if a cell takes 24 hours to divide, it means the mitotic division takes place in 1 hour and the rest 23 hours will be taken for the preparation. It simply means when a cell divides, 95% of its time has been covered under the interphase and 5% time have been consumed by the M phase. Now the interphase is further divided into three phases. First phase is called as G1 phase, which is called as gap. Gap means preparatory phase. Second is S phase. In S phase basically what happens, the synthesis takes place. Synthesis of DNA, which plays the major role during the division. And next is called as G2, which is again a gap. These two gaps means the cell prepare itself for the division. First gap means cell prepare itself for the synthesis of DNA, and second gap means the cell prepare itself for the mitotic phase. Now we will do it in a now let's see that this is the cell cycle. Cell cycle consists of four phases. One is G1, S, G2 and M phase. Now this is called as interphase, means when the cell prepare itself, which is the most time consuming phase. In this, first we will study about the G1 phase, what happens during this. So we can say this is the major preparatory phase, or we can also say that this is a gap between the G1 and the S phase. This is the major time which will be taken by the cell. Now what happens during this phase? First, this is the preparatory phase, means it is preparing itself for the DNA replication. So whatever the things are needed for the DNA replication, all these things will be prepared in this phase. So number one is protein synthesis. Now why protein is required? The requirement of protein is for the enzymes. All the enzymes are made up of protein. Now why enzymes are required? Enzymes are required for the DNA replication. When one DNA strand is there and it has to form the another strand for the replication. Replication means replicate. One strand will form the second strand. So at that time enzymes like topoisomerase, all these enzymes are required. So protein synthesis is must. Second is RNA synthesis. Now why RNA synthesis is required? Because according to the total process, when the proteins are formed, proteins will be formed only when RNA will be present. So RNA amount will increase. Next step is ATP. ATP synthesis. ATP are always called as energy currency. So energy is always required for the whole procedure. Second, raw material required for the DNA replication. Now, DNA is formed of pentose sugar. So, pentose sugar is required. Phosphoric acid is required. Nucleotides are required. All these things will be prepared during the G1 phase because of the formation of DNA replication. Now, the cell organelles duplicate. Why the cell organelles will duplicate? Because in mitosis, we know that one cell divides to give rise to two daughter cells. If one cell contains 46 chromosomes, then equal amount of chromosomes will be given to the two daughter cells. If equal amount of chromosomes are given to the two daughter cell, it means both the cells are clone of the parent cell. So, for cloning, every organelle needs to be in equal amount. If parent cell contain 10 mitochondria, 10 Golgi bodies, it means though these two cells must contain 10 Golgi bodies and 10 mitochondria. So, that is why to divide equally, the number of organelles will be doubled. So, it will duplicate. Now, if all the things are increasing, if energy is required, protein is increasing, cell organelles are increasing, it itself means the cell size is also increasing. If the cell size is increasing, it means the cell growth has taken place. So, all these are the steps which takes place during the gap 1. Now, this was the preparation for the synthesis phase. Now, this is the major event. Now, let us see what happens during the S phase. Here, the DNA will replicate. Now, the question arises, why DNA replicates? What is the need of DNA replication during this cell cycle? As I have already discussed, number of chromosomes are present 46. For example, I am taking example of human cell. 46 number of chromosomes are present in the parent cell and equal amount has to be distributed in the daughter cell. So, that is why DNA will replicate, DNA will double in amount. The major thing which we need to learn here, that double amount of DNA will takes place. Now, the question arises and which you have to take in consideration, that only the amount of DNA will increase, not the amount of chromosome. The amount of DNA is always considered as C. So, then the amount of the content of the DNA will become 2C. N is considered as the number of chromosome, it will remain same as N. Let us take example, if I am drawing here, this is a chromosome. Now, the number will remain same, the amount of chromosome will remain same, only what will happen, it will form its sister chromatid. The blue one is sister chromatid and red one was the parent chromatid. Now, the number of chromosome, here it is one chromosome, this one is the second chromosome, here it is also considered as one chromosome and this will be considered as the second chromosome. The amount of DNA has increased, but the amount of the number of chromosome will remain same. So, we will write here number of chromosome remain same. This is a major question which is being asked in the question in your paper and as well as related to the NEET. Number of chromosome will remain same. Only what will happen? The content, or you can say volume of DNA gets double. Now, the second is centrioles duplicate. Centrioles will get the double, and what are centrioles? Which are required during the cell division, and these gets double or duplicate only in the animal cell. You have to keep this point again in your mind, that it will duplicate only in the animal cell, not in plant cell, because in plant cell division takes place through the cell plate. So, no centriole is required. Now, the third is histone proteins will increase. In few books it is mentioned that histone proteins gets increased in the G1 phase, but in few books it is mentioned that histone proteins are increased in the S phase. Now, what is the difference between these two? In lower eukaryotes, 50% of histone protein is being synthesized in the G1 phase and 50% is being synthesized in the S phase. But in the higher eukaryotes, all the histone protein is being synthesized in the S phase, though that's why we always mention the increase of histone proteins in the S phase. Now our second last point is G2 phase here. Now this phase is basically called when the cell prepare itself for the division, because after this M phase will start and M phase is called as the division phase. So here the cell will prepare itself for the division. So what will happen? All these contents will start again here. Again, now the protein synthesis will increase. Now what is the need of protein here? Here protein will increase for the formation of tubulin protein. Now why tubulin is required? It is required for the formation of spindle fibers. What are spindle fibers? When a cell is dividing, two astral rays are formed and here the spindle fibers will be formed where the chromosome will set themselves. So these spindle fibers are made up of protein tubulin. So here protein synthesis is required. Now the next is plasma membrane will increase. Now what is the need of plasma membrane? When the new organelles are formed, because as we have already discussed, the mitochondria will again form, Golgi bodies, all the organelles will form again. So plasma membrane is also required for the formation of the organelles, their outer membrane. Now again third point is synthesis of ATP. Why ATP is required? When the chromosome needs to move from equator to pole, they require energy because the astral rays will pull them towards the polar side. So for that they require energy for the movement from the equatorial plate to the polar region to the polar side. So energy is required. So we can say for the movement of chromosomes, energy is required. And in the last RNA synthesis, it is all again required for the, because all the three type of RNA, tRNA, mRNA and ribosomal rRNA, these are required for the functioning of the whole process. So these three are the phases: gap 1, S phase and G2 phase, and here the interphase ends, and from here the basic division phase will start, which is called as M phase, and we will do that in our next session. This is a very quick review for your cell cycle. You can make your cell cycle in this way, or either you can write all the phases in the cell cycle in your books, so that it will be a quick revision for you. Thank you.